today we will start with our chapter number 5, that is Principles of Inheritance and Variation, and this is chapter number 5 for class plus 2. And today we will start with our part number 2. In part number 1 we will discuss up to the law of independent assortment. And as all we know that heredity is the transmission of characters from parents to the offsprings from one generation to the next generation. And in the law of independent assortment, it states that it separates the alleles from different traits of parents to the offsprings. And Mendel's used dihybrid cross, that is the cross between the two different traits, in order to explain the law of independent assortment. So this is all about up to the part number 1. And let us start with our part number 2, that is the inheritance of one gene. So what do you mean by the inheritance of one gene? Inheritance of one gene can be explained using Mendel. So inheritance of one gene concept, that is to be explained by Mendel, one of the hybridization experiment. And that concept is explained on the basis of the hybridization experiment, such as crossing, and that hybridization experiment is explained on the basis of crossing between the tall and dwarf plant. That means there is a crossing between the tall plant as well as dwarf plant. When the tall and the dwarf plants are crossed, जब हम tall plants को और dwarf plants को cross करेंगे, that means there is a crossing between the tall plants and the dwarf plants, the resulting first hybrid generation, जो वहां से जब हम crossing करेंगे between the tall plants and the dwarf plants, उसके बाद जो हमारी first hybrid generation बनेगी, that first hybrid generation is known as the filial progeny. That is known as the filial progeny or the first generation progeny. That means जो हमारी first hybrid generation बनेगी, that hybrid generation is known as the first progeny or the first generation progeny. Mendel absorbed that. अब उसके बाद Mendel ने क्या observe किया? Mendel absorbed that all the plants formed are tall. जब उसने crossing की between the tall plants and the dwarf plants के बीच में, तो उसमें जो उनकी फर्स्ट फीलियर जनरेशन या फर्स्ट प्रोजेनी जनरेशन जो बनी, उसमें उन्होंने ऑब्जर्व किया कि ऑल द प्लांट्स आर टॉल, जितने भी प्लांट्स बने ऑल दीज आर टॉल. देन द F1 प्लांट्स वर सेल्फ पॉलिनेटेड. इसका मतलब ये हो गया कि जितने भी हमारे F1 प्लांट्स थे, ऑल द F1 प्लांट्स वर सेल्फ पॉलिनेटेड एंड दे प्रोड्यूस द F2 जनरेशन. और जितने भी हमारे प्लांट्स थे वो सेल्फ पॉलिनेटेड थे. Self-pollinated means they produce the F2 generation. The progeny obtained has 3 tall plants. अब जो progeny बनी, that means that progeny has 3 tall plants and 1 dwarf. That means ratio is 3:1. तो इसमें जो हमारी इसमें जो progeny बनी, that progeny obtained, that means 3 plants are tall and 1 plant is dwarf. That means the resulting ratio is 3:1. So this is the concept regarding the inheritance of one gene. After that, the next law, that is the incomplete dominance. Next important law regarding the principles of inheritance and variation is the incomplete dominance. So what do you mean by incomplete dominance? When the dominant allele is not completely dominant. जब हमारा dominant allele जो होगा, that is not completely dominant. जब completely dominant नहीं होगा over the recessive allele. When the dominant allele is not completely dominant over the recessive allele and the F1 hybrid forms are intermediate. और जो हमारे F1 hybrids होंगे, they are intermediate of the two parents. That means जो हमारे F1 hybrids जो बने, F1 hybrids forms, they are intermediate of the two parents, and that phenomenon is known as the incomplete dominance. That phenomenon is known as incomplete dominance. The figure given below, अब जो हम नीचे figure लगाएंगे, that given below explains when the red flower, जब हम red flowers को, that are dominant flowers, were crossed with the white flower. जब हम red flowers को crossed करेंगे white flowers के साथ, and red flowers are dominant and the white flowers are recessive, then the F1 generation contains flower. जब हमारी F1 generation जो बनेगी, that F1 generation contains the flowers, and that flowers are pink in color. That means that flowers, they are pink in color. They are intermediate between the red flowers as well as the white flowers, and the flowers that form, they are pink in color. So this is the diagrammatically representation of the incomplete dominance. So this is the F1 generation, and all the flowers, they are red and the white flowers. That means जब हम रेड और व्हाइट फ्लावर्स को क्या करेंगे, क्रॉस करेंगे, देयर इज अ क्रॉसिंग बिटवीन द रेड एंड द व्हाइट फ्लावर्स, एंड द रेड फ्लावर्स आर डोमिनेंट एंड द व्हाइट फ्लावर्स आर द रिसेसिव. तो जो F1 जनरेशन बनेगी, दैट इज पिंक इन कलर. तो उसमें जो फ्लावर्स बनेंगे आफ्टर द क्रॉसिंग ऑफ द रेड फ्लावर्स एंड द व्हाइट फ्लावर्स, द फॉलोइंग कलर फॉर्म दैट इज पिंक इन कलर. वो कैसा बनेगा, हमारा पिंक कलर का बनेगा, एंड दैट जनरेशन इज नोन एज द F1 generation. So these there is a crossing between the red flowers and the pink white flowers. So these are the sperms, that is CR, and this is the CW, and these are the eggs. जब हम इनकी क्रॉसिंग करेंगे तो this is the red colored flower, that is CR, and this is the pink flower, that is CR and CW, and this is also pink flower, that is CW. That means जब हम क्रॉसिंग करेंगे आपस में, and CW, that is the white flower. So this is the F2 generation. और जो रेशियो बनेगा, that is red color जो है हमारे पास, that is 1, और pink flower जो है, that is the 2 in number, तो 1 ratio, 2 ratio, and white flower is 1. That means जो हमारे पास रेशियो बना, that is 1 ratio 2 ratio 1, और 1 रेशियो के जो हमारे रेड कलर जो है, that is CR, एंड पिंक कलर जो है, that is CW जो है, that is 2 in number, and CW that are white in color. तो रेशियो जो बनेगी हमारी इस इनकम्प्लीट डोमिनेंस में, दैट इज 1 ratio 2 ratio 1. तो इसमें इनकम्प्लीट डोमिनेंस में हमने ये चीज देखी कि जब हम क्रॉसिंग करेंगे रेड फ्लावर्स और व्हाइट फ्लावर के बीच में, तो रेड फ्लावर्स दैट आर डोमिनेंट एंड व्हाइट फ्लावर्स दे आर रिसेसिव. तो जो उनसे हमारी F1 जनरेशन बनेगी, दैट मींस द फॉर्म्ड फ्लावर्स दे आर पिंक इन कलर. तो जो हमारी उसे जनरेशन बनी, दैट इज पिंक कलर. तो रेशियो जो बना हमारा, दैट इज 1 ratio 2 ratio 1. तो जो रेड कलर के थे, दैट इज 1, एंड जो हमारे पिंक कलर के बने, दैट इज 2, एंड जो व्हाइट कलर के बने, दैट इज 1. तो रेशियो दैट इज 1 ratio 2 ratio 1. सो नेक्स्ट इज द, फॉर एग्जांपल जो था, दैट इज flower color in Mirabilis jalpa, that is for a clock plant, and the phenotypic and the genotypic ratio, जो उनके बीच में phenotypic ratio as well as genotypic ratio जो बना, that is 1 ratio 2 ratio 1. So this is the example of the incomplete dominance. Our next important topic regarding this chapter, that is the multiple allelism or codominance. Multiple allelism as well as codominance. When a gene exists in more than 2 allelic forms, जब हमारा कोई भी जीन जो है, that is exist in more than 2 allelic forms, तो जब वो 2 से अधिक allelic forms में exist करेगा, that phenomenon is known as multiple allelism. That concept is known as multiple allelism. For example, multiple alleles are the inheritance of A, B and O blood groups. फॉर एग्जाम्पल हम ये चीज कहेंगे कि जो मल्टीपल एलील्स हैं, दैट मीन्स द मल्टीपल एलील्स, द इनहेरिटेंस ऑफ ए, बी एंड ओ ब्लड ग्रुप्स इन ह्यूमन बींग्स. दैट मीन्स जो मेन ब्लड ग्रुप्स ह्यूमन बींग्स में एग्जिस्ट करते हैं, दैट दे आर ए, बी एंड ओ. द जीन्स फॉर ब्लड ग्रुप्स अकर, अब जो जीन्स हमारे ब्लड ग्रुप्स में अकर करेंगे इन मल्टीपल इन 3 एलीलिक फॉर्म्स. दैट मीन्स जो हमारे ब्लड ग्रुप्स जो है, या जीन्स जो हमारे ब्लड ग्रुप्स के लिए, द जीन्स फॉर द ब्लड ग्रुप्स अकर इन 3 एलीलिक फॉर्म्स, वो 3 एलीलिक फॉर्म्स में एग्जिस्ट करेंगे, दैट इज IA, IB एंड I. IA, IB and I. An individual can possesses any 2 of these alleles. कोई भी इंडिविजुअल जो है इन 3 एलील्स में से कोई भी 2 एलील्स जो होंगे वो पोजेस करेगा. दैट मीन्स दे कैन पोजेस एनी 2 ऑफ द एलील्स. द जीन IA कोड्स फॉर ग्लाइकोप्रोटीन. जो हमारा जीन IA है, दैट कोड्स फॉर द ग्लाइकोप्रोटीन A which is responsible for A blood group. That means this gene IA codes, that is for glycoprotein A, and that is responsible for A blood group in human beings. And next is the gene IB codes, that is for the glycoprotein B, and which is responsible for the blood group B in the human beings. That means हमें ये हो गया कि जो gene IA code है, that is for glycoprotein A and it is responsible for the blood group A in the human beings, whereas the gene IB codes for the glycoprotein B, that is responsible for the blood group B in the human beings. Next is this is the graphic representation of the blood type as well as the genotype. If the blood type is A, that means the genotype, that is IA, IO, IA, IA, and the genotype is AO as well as AA. If the blood group is B, that means the genotypes are IB, IO, IB, IB, and the genotypes further are BO and BB. Next, if the blood group is AB type, अगर blood group AB type है, that means the genotypes are IA, IB, IO, IO, and further they have genotypes AB. And if the blood type is O, that the genotypes are IO, IO, and the further genotype, that is the OO. So this is the multiple allelism in the blood groups. So this is the diagrammatically or the graphic representation regarding the multiple allelism in the blood groups. Next is the genes I. The genes I do not produce any glycoprotein. अभी हमने देखा कि जो हमारे genes हैं IA and IB, that are responsible for the glycoprotein A and glycoprotein B, and they produce the blood group A and B, whereas gene I, they do not produce any type of the glycoprotein, and so the person who is homozygous, तो इसका मतलब ये हो गया कि जो person homozygous है for it will have O blood group, तो उसमें जो blood group होगा that is O type. And genes IA and IB, both the genes that are IA and IB are dominant over I. अब ये both the genes that are IA and IB, they are dominant over the I. IA and IB alleles are equally dominant. अब जो IA as well as IB genes हैं, they are equally dominant. दोनों के दोनों जो हैं, they are dominant and produce both the glycoproteins A and glycoprotein B. And the blood groups, that is AB. तो इनमें जो blood group होंगे that is AB type. Such alleles are known as, ऐसे alleles को हम क्या कहेंगे? Co-dominant alleles. Such type of the alleles, they are known as the co-dominant alleles. So this is all about the multiple allelism in the blood groups. Next is the inheritance of the two genes. What do you mean by inheritance of the two genes? Mendel also worked with the pea plant. जैसे कि हम सभी जानते हैं कि Mendel has to be worked on the pea plant, that is the Paytm sativa. So Mendel also worked with the pea plants that differ in two characters, and that pea plant, it is differ in the two characters. He had chosen the color. अब उसने जो choose किया, that means they have to be choose the color and the shape of the seed. He had to be choose the color as well as the shape of the seed to explain the inheritance of the two genes. अब उन्होंने क्या कहा कि he had chosen the two characters, especially the color as well as the shape of the seed, to explain the inheritance of the two genes. Y represents the dominant yellow. जो Y character था, that represents the dominant yellow color seeds color, whereas small y represents the recessive green color. Capital R represents round shape of the seed. जो capital R था, that represents the round shape of the seed, whereas small r represents the wrinkled shape of the seed. The genotypes of the parents can, after that, the genotypes of the parents can be written as capital RR, capital YY, and small rr, small yy. That means जो genotypes होंगे parents के that can be represented in the form of capital RR, capital YY and small rr, small YY. The gametes after that, उनके बाद जो उनके gametes बनेंगे, that means the gametes are capital R, capital Y and small r, small y. Unite, आपस में वो unite करेंगे on the behalf of the process that is fertilization and to produce the F1 hybrids. जब जो gametes आपस में unite करेंगे on the behalf of the process that is fertilization, they produce the F1 hybrid that is capital R, small r, capital Y, small I, small y. So this is the F1 hybrid that is produced after the unitation of the gametes, that is the capital R, capital Y and small r, small y. Inheritance of the two genes inheritance of the two genes was also used to explain. अगर हम inheritance of the two genes देखें, that was also used to explain the law of independent assortment based on the crosses. अगर हम crosses के behalf पर देखें, the F2 ratio was found to be. अगर हम crosses का behalf पर देखते हैं, on the behalf of the crosses, the F2 ratio, जो F2 ratio बनेगी, that was found to be 9:3:3:1. So this is the F2 ratio that was found, that is 9:3:3:1. So this is the dihybrid cross. So these are the capital YY, capital RR seeds crossing with small yy, small rr, that is the parental generation. जब इनकी क्रॉसिंग करेंगे, after the crossing the F1 generation form, and that F1 generation is capital Y, small y, capital R, small r. After that the phenotypes formed, that is gametes formed from the heterozygous parents. अब जो gametes बनेंगे and that gametes formed from the heterozygous parents are capital Y, capital R, small y, capital R, capital Y, small r, small y, small r. And these are the gametes from the heterozygous parents, and from this side these are the gametes from the heterozygous parents, that is capital Y, capital R, small y, capital R, capital Y, small r and small y, small r. After the crossing between the gametes of the heterozygous parents, that is capital YY, capital RR, capital Y, small y, capital R, capital R, capital Y, capital Y, capital R, small r. Next, capital Y, small y, capital R, small r. Next, small y, capital R, capital R. Small y, capital R, capital R. Small y, capital R, small r. Next, capital small y, capital R, small r. Next, capital Y, capital R, small r, capital Y, small y, capital R, small r. Capital Y, small r, small r. Capital Y, small r, small r. Next, capital Y, small y, capital R, small r. Capital small y, capital R, small r. Next, small y, small r, small r. Small y, small r, small r. So this is the inheritance of the two genes, that is the dihybrid cross between the two parents, that is the capital YY, capital RR and small yy, small rr. That means जो ratio बना इसमें हमारा, तो ratio जो बना, that is the 9:3:3:1. And these are the phenotypes that are formed after the dihybrid cross between the two parents, and the outcoming ratio is found to be 9:3:3:1. Next is the chromosomal theory of inheritance. Next important theory regarding the inheritance of variation is the chromosomal theory of inheritance. Chromosomal theory of inheritance is also known as Boveri's certain chromosome theory. This theory is also named as Boveri's certain chromosome theory. And according to this theory, the main characteristics of this theory are genes are located at specific location on the chromosomes. That means जितने भी genes थे, they are located on the specific location on the chromosomes. Homologous chromosomes separate during meiosis. जितने भी homologous chromosomes थे, they are to be separate out during the process of meiosis. Fertilization restores chromosomes. Fertilization की process जो थी वो क्या restore कर रही थी? Chromosome numbers को to diploid condition. Chromosomes segregate as well as assort independently. जो भी chromosomes थे, they can be segregate as well as assort independently. So this is the chromosomal theory of inheritance. Next is linkage and recombination. What do you mean by the process of linkage as well as recombination? T.H. Morgan conducted different experiments. जो scientist थे, that is T.H. Morgan, different experiments उन्होंने जो conduct किए थे, they conduct different experiments to understand the process of linkage. That means उन scientists ने different experiments को conduct किया to understand the process of the linkage as well as recombination. He carried out, उन्होंने क्या carried out किया? Several dyed hybrid crosses in Drosophila. उन्होंने several crosses जो थे, उन्होंने several dyed hybrid crosses जो थे उनको study किया in Drosophila to study the genes that were sex linked. जो कि gene जो थे वो क्या थे? Sex linked थे. For example, Morgan hybridized yellow-bodied white-eyed females to brown-bodied, red-eyed males and intercrossed those F1 progeny. He observed that two genes, उन्होंने क्या observe किया कि जो दो genes हैं, when do not segregate independently, जो दो genes हैं, the two genes do not segregate independently of each other and F2 ratio deviated very significantly, और जो F2 ratio था, that is to be deviated very significantly from the 9 ratio 3 ratio 3 ratio 1. That is expected when the two genes are independent. जिसमें कि एक expect किया गया था कि when the two genes are to be independent. This leads to the conclusion. इससे क्या conclusion निकला? This leads to the conclusion that genes are linked. इससे ये conclusion निकला कि जो दो genes थे, they are to be linked with each other. Such genes which are physically, ऐसे genes जो कि physically क्या हैं, linked करते हैं एक दूसरे के साथ, this process is known as linkage. Recombination. After the linkage, the next process is the recombination. That means recombination is defined as recombination. को हम कैसे define करेंगे? That is rearrangement of the genetic material. Recombination के process में there is the arrangement of the genetic material. For example, by crossing over is known as recombination. जब हम crossing over करेंगे, by crossing over is known as the recombination. It is responsible for the formation of recombinants. That means recombination is responsible for the formation of recombinants. That is progeny formed by the combination of the two parents. जो progeny बनेगी, that progeny is formed by the combination of the two parents. That is, it is responsible for the variation. That means that process is responsible for the variation. So this is the diagrammatically representation regarding the variation, that is wild type and the black vestigial. That is BVG, small b, small vg. Crossing करेंगे. These are the parental genotypes and these are the recombinant genotypes. जब भी crossing करेंगे, that means they have the characters of the wild type and these are the characters of the black vestigial. That is small v, small vg, small b and small bg. These are the characters of the black vestigial and wild type. These are capital B, small g, small b, small bg. Parental genotypes are small b, small vg, capital B, capital B, small g. Recombinant genotypes are capital B, small vg. Next, recombinant genotype small b, capital B, small g, small b, g. After that, parental genotypes b, small vg से जो बनेंगे, that is b, small bg, b, small vg. That is the black vestigial. Next is BBG वाले जो है, that is capital B, small vg, small b, small vg, that are wild type. That is 965. नेक्स्ट स्मॉल बी, कैपिटल बी, स्मॉल बीजी के जो है दैट इज ग्रे वेस्टिजियल है, स्मॉल बीजी दैट इज 206. स्मॉल बी, कैपिटल बी, स्मॉल बीजी दैट आर द ब्लैक नॉर्मल, दैट इज 185. सो दीज आर द black vestigial and white type. These are the parental phenotypes, whereas gray vestigial and black normal, that are 206, they are gray vestigial and black normal, that are 185. These are the recombinant phenotypes. So this is the diagrammatically representation of the linkage and the variation in the Drosophila. Next is the sex determination. How can you determine the sex in the living organisms? Sex determination is a system that determines the development. तो sex determination जो है, that is a system that determines the development of the sexual characteristics in an organism. Different organisms has different type of the sex determination. That means all the organisms, they have to be different types of the sex determination. That means each and every organisms, they have different types of the sex determination in them. In insects, the mechanism of sex determination is of exotype. अगर हम insects के केस में देखते हैं तो जो sex determination है that is in the form of exotype, and in this eggs have X chromosome. तो इसमें हम जो देख रहे हैं exo, that means जो organisms हैं उनमें जो eggs होंगे that are they have X chromosomes. उनमें जो chromosomes होंगे they have X chromosomes. But sperms may have 1 X chromosome. पर जो sperms हैं उसमें भी 1 X chromosomes होंगे. Some do not have any X chromosomes. पर कुछ ऐसे भी होंगे they do not have any type of the X chromosome. As X chromosome is determining the sex of an individual. अगर हम ये कह रहे हैं कि as X is the determining of the sex of any organism or any individual, X chromosome is known as. तो उस यहाँ पर हम ये कह सकते हैं कि X chromosome is known as the sex chromosome. अगर कोई भी X जो क्रोमोसोम है वही डिटरमाइन कर रहा है किसी भी ऑर्गेनिज्म के सेक्स को, तो हम उस यहाँ पर ये कह सकते हैं कि X chromosome is also known as sex chromosome. Chromosomes other than the sex chromosome. अगर chromosomes other than the sex chromosomes is known as autosomes. Next concept is what do we mean by autosomes? That is chromosomes other than the sex chromosomes. They are known as the autosomes. Mammals including man. Mammals including man has XXXY type of the sex determination. अगर हम mammals के केस में देखते हैं such as human beings, that means they have XX and XY type of the sex determination. Females carry X chromosomes in their eggs, whereas males they have to be carry Y chromosomes. जबकि जो males जो है either उनमें X chromosome भी हो सकता है या Y chromosome भी हो सकता है, whereas in case of the females they have only X chromosome, whereas in case of the males either they have X chromosome or Y type of the chromosomes. That is why females are said to be homogametic. इस वजह से females को क्या कहा जाता है? Homogametic. Homogametic means same type of the gametes. यहां पर आपका क्वेश्चन रेज हो सकता है, दैट इज वेरी इंपॉर्टेंट फ्रॉम द एग्जामिनेशन पॉइंट ऑफ व्यू. दैट इज व्हाई द फीमेल्स दे आर सेड टू बी होमोगैमेटिक. दैट मीन्स द फीमेल्स दे हैव टू बी सेड होमोगैमेटिक बिकॉज दे हैव ओनली वन टाइप ऑफ द सेक्स क्रोमोसोम, दैट इज X chromosome. On the behalf of that reason, females they are said to be homogametic, and homogametic means that they have same type of the gametes. and males to be heterogametic, और males को हम heterogametic बोलते हैं because they have different types of the gametes. Either they have X type of the chromosomes or Y type of the chromosomes. On the behalf of that reason, they are said to be heterogametic, or they have different types of the gametes. Next is, this is the diagrammatically representation of the different types of the sex determination in different types of the organisms. So this is the, in case of the mammals, that is the human beings, this is the female and this is the male. And in females, the number of chromosomes, sex chromosomes is 44 plus XX, whereas in case of the males, that is 44 plus XY. This is the birds, that is 76 plus ZW, whereas 76 plus ZZ. So next is the, these are the insects, that is 6 plus XX, 6 plus XY, or 6 plus XO. Next is, that is in case of grasshopper, 22 plus XX, 22 plus X. So this is the different types of the sex determination in the different types of the organisms. So next is the mutation. Next topic of this chapter is the mutation. So what do you mean by mutation? क्या समझते हैं आपकी mutation क्या है? Any change in the DNA sequence. DNA sequence में किसी भी change का आना, any change in the DNA sequence is known as mutation. That means DNA sequence में जैसे ही change आता है, किसी भी type का change आना DNA sequence में, वो क्या कहलाता है? Mutation. It causes a heritable change in the DNA. अब mutation क्या cause करता है? Heritable change in the DNA. The genotype as well as phenotype will be affected due to mutation. Both the genotypes as well as phenotypes, both have to be affected by the process of mutation. There are different types of the mutations. यहाँ पर different types के mutations हैं, such as frame shift mutations, deletions, insertions, substitutions, duplications, etc. Are some mutations are harmful but some are not harmful. तो यहाँ पर various types के mutations हैं जो occur करते हैं. That are the frame shift mutations है, deletions है, insertions है, substitutions है, duplications है. So there are many types of mutations that occurred, but out of them some mutations are harmful and some mutations are not harmful. So next is the frame shift mutations causes. अब ये जो different types के mutations हैं, how can we say that some are harmful and some are not harmful? So frame shift mutations causes loss or addition of DNA bases. अब frame shift mutation जो है वो क्या cause करता है? They cause the loss of the DNA और addition of the DNA bases. वो DNA bases का loss भी कर सकते हैं और addition भी कर सकते हैं, which changes the reading frame. और जैसे ही उसमें addition होगा DNA basis का, ये loss होगा DNA basis का, तो उससे क्या होगा? Changes the reading frame. Addition of DNA bases is known as insertions. Addition of DNA bases. DNA bases की addition होना जो है वो क्या कहलाएगा? Insertions. Removal of DNA bases. DNA bases का remove होना, that means that is known as deletion. उसको हम क्या कहेंगे? Deletions. A piece of DNA if copied more than one time. अगर कोई भी DNA piece है, अगर उसकी copied करते हैं हम more than one time, एक समय से ज्यादा अगर हम उसकी कॉपिंग करते हैं, that is known as duplication. तो duplication क्या हो गया कि a piece of DNA copying if copied more than one time, अगर हम किसी भी DNA piece की एक समय से ज्यादा अगर हम उसकी कॉपिंग करते हैं, more than one time we can copy the DNA piece, that is known as duplications. Thus these mutations changes the DNA sequence. इस यहाँ पर हम ये कह सकते हैं कि जो DNA sequence होती है या mutations जो होती हैं, that means mutations they can changes the DNA sequence. तो जो mutations होते हैं वो क्या change कर देते हैं? DNA sequence को, या DNA sequence होती है that can be changed on the behalf of mutations is known mutations which ultimately leads to the formation of wrong protein. तो उसकी जो अल्टीमेटली जो फॉर्मेशन है वो किस चीज की फॉर्मेशन करता है, रॉन्ग प्रोटीन की। नेक्स्ट इज जेनेटिक डिसऑर्डर्स। देयर आर many types of the genetic disorders that occurs. First genetic disorder is the pedigree analysis. Charts used to understand the inheritance pattern. जिन charts के बिहाव पर हम किस चीज को understand करते हैं, inheritance pattern in a family is known as pedigree. उसको हम क्या कहेंगे, pedigree. It is also used to find out the genetic disorders in the family also. इस बिहाव पर हम किसी भी family में जो genetic disorders हैं उनको भी हम find out कर सकते हैं। All the family members are mapped in the form of a tree. जितने भी family members हैं and they are mapped in the form of a tree. So this is the genetic disorder, and the first genetic disorder is the pedigree analysis. Next is the Mendelian disorders. Genetic disorders are grouped into two categories. अब genetic disorders को we have to be categorized into two different categories, that is the Mendelian disorder and the chromosomal disorders. That is the Mendelian disorders. They have to be categorized into two categories. First is the Mendelian disorder and the second is the chromosomal disorders. Mendelian disorders are based on the alteration or mutation in the single gene. तो जो Mendelian disorders हैं, they are based on the alteration or mutations in the single gene. Whereas most common of these are hemophilia. उनमें से सबसे most important common disorder जो है that is the hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. Such Mendelian disorders can be dominant or recessive. तो different types के Mendelian disorders हैं and these Mendelian disorders are hemophilia, sickle cell anemia, phenylketonuria and color blindness and next is the thalassemia. तो ये disorders जो हैं, these disorders they can be either dominant or recessive. जरूरी नहीं है कि वो dominant ही है या जरूरी नहीं है कि वो recessive है. Either they can be dominant or recessive. Can also be linked to sex chromosomes. तो ये जो हमारे disorders हैं, they can be linked to the sex chromosomes such as hemophilia and color blindness. Next important genetic disorder, first is the pedigree disorder, second is the Mendelian disorder, and after the Mendelian disorder, the next important genetic disorder is the chromosomal disorders. When disorders are caused due to absence or excess, जब कोई भी disorder जो है वो caused होता है due to the excess or abnormal arrangement, मतलब जरूरत से ज्यादा होना या फिर abnormal arrangement का होना of one or more chromosomes, एक या एक से अधिक क्रोमोसोम का जरूरत से ज्यादा होना या abnormal arrangement होना, वो क्या कहलाता है, chromosomal disorder. Improper segregation of chromatin during cell division cycle results in the gain or loss of chromosomes called aneuploidy. उसको हम क्या कहते हैं, aneuploidy. For example, Down syndromes result in the gain of extra copy of chromosome. अगर हम Down syndrome की बात करते हैं तो वो result करता है gain of extra copy. मतलब जरूरत से ज्यादा किस चीज का gain करना, extra copy of the chromosomes. Gain of extra copy of the chromosome 21. Similarly, Turner syndrome, अगर हम Turner syndrome की बात करते हैं, results, अब Turner syndrome किस वजह से result करता है, due to the loss of an X chromosome in human female. अब Turner syndrome जो है वो किस वजह से हो रहा है, due to the loss, किस चीज का loss होना है, X chromosome का in case of the females, human females. Absence of cytokinins, अगर cytokinins की absence है after telophase stage of cell division, that results in an increase in a whole set of chromosome in an organism, and this phenomenon is known as polyploidy. So this condition is often seen in plants. जरूरी नहीं है कि that condition is possible only in case of the human beings, but this case is also possible in case of the plants. So this is all about this chapter, that is the chapter number 5, Principles of Inheritance and Variation, and this chapter is for class plus 2, and this chapter is completed in two parts, that is the part number 1 and part number 2, and all the concepts are to be cleared in both the parts, that is part number 1 and part number 2. Next we will continue our next lecture with our next chapter, that is chapter number 6.